during the lab classes, we will have one block on the genotyping method and methods, and one on the molecular diagnostics of Staphylococcus infections. And this will be, these classes are going to use the techniques like RNA isolation, reverse transcription, and qPCR. So as you see, there are 4 main blocks. We are meeting 6 times, and certain blocks of experimentations will take part during several classes. So please make sure that you are that you know what we are going to do and when, and I will send you this schedule of the labs and task during the labs later on. Okay, so how you should prepare? So first of all, you bring clean lab coat, instructions, notebook, and a pen. We do not allow any cell phones and calculators during the lab classes. The only exception is if you have someone sick and you need to have the cell phone with you, we kindly ask you, to put it, in the task, in the pocket, somewhere. Please do not keep the cell phone on the laboratory benches, okay? You please leave the jackets at the reception, and if it comes to the absence, of course, if you are sick and something happens to you, please inform us, send the doctor release form so we can justify your absence. Of course, we are now during the pandemic period, so of course you come to the classes when you are healthy. Even if you are vaccinated, it does not prevent you 100% from contracting Delta variant, see me, for example. So we kindly ask you to come healthy. It means without fever and any symptoms of cold or flu. We do not know actually that if you are going to be sick during, I don't know, several weeks, because of the complications of whatever, we will make the effort for you so you can actually make this course done, but we will take these particular cases one by one. If you see the symptoms of coronavirus infection, please inform us immediately, okay, and perform a test. Important thing is, remember that on the entry to the every classes you will sign a declaration that you are not showing any symptoms of the disease. So how you prepare for the classes? All the materials that you need to know and all of the topics have been already told you during the lectures or are going to be told soon. You will have instructions, and then, of course, important thing, all the calculations that are in the instructions should be done before. So read also all instruction and know and understand what step by step we are going to do and why during the classes. So now the question is, could you please check? There was a problem with receiving the material for the instructions for the lab class, the first lab class, which has been sent through the students portal. Could you please verify now that you have received it, and please let us know that you have it or don't? Could you please do it now?
Okay.
It's okay, you have it?
Yeah, we have it.
Okay, good. So, grading, on what you are going to be graded and how. So your grading from the lab classes will compose from 50% of the grade will be the mean from 3 entry tests that you are going to take. 25% from reports, there will be 3 reports to be sent, And the last 25%, it is so-called performance, which is the how you keep the safety measures, how you organize your time and space, and it will be also on your activity. So there are 3 entry tests. However, we meet several times, and different topics are going to be covered, and all of the leaders of the groups are going to ask you questions during the classes. So please be aware of it, that you are judged also on your activity. So entry test, as I told you, there are 3 tests at the first lab, at 4th, and at 5th. Each entry test will have 3 or 4 questions, sometimes maybe 5. And then there will be also calculations to be performed. All topics that you need to prepare are given at the top of each instruction, so please read carefully and prepare. We allow 1 unsatisfactory mark, okay? So if you are not well prepared, or you had problems with answering questions and you did not perform well, then only 1 unsatisfactory mark you can have. If you have more, which sometimes unfortunately happens, is you will have to write a correctional exam at the end at the end of the all classes. And here you have the example of topics to prepare, how they are given to you. So they are all necessary things listed, what you need to prepare for each class. Then we go to reports. 3 reports to be given to after classes. First report you give us after lab second meeting in the laboratory, and it will be on the DNA isolation and analysis. The second report will be after our 4th meeting, and it will be on the interferon lambda genotyping. And then the last report will be after lab 6, not 5, sorry for the mistake, and it will be the report on the RNA isolation and reverse transcription and qPCR. This report you will send actually in January after your second online meeting. On the second online meeting, what you are going to do, you are going to analyze the results of the qPCR with the lecturer. So then the results are analyzed, and you know how to actually read the results and how to write and calculate the relative expression of the gene. Okay, so this is about the reports. We also allow you to correct first report. We know that actually it's the one which is the most difficult for you, because it's the first one, and everyone has every laboratory classes on different subject has different requirements, so you can correct first report. We will also allot the time, and it has been allotted on the lab 3 after you have given us the first report, so we can actually discuss with you in detail on this first report. And how the reports are going to be prepared. They have to be sent by mail at the due time. So if the leader asks you to send by noon on the specific day, please do so. Okay, but be careful. Very often, some of the mails finish in the spam. It's not even those ones which has been given by the university. So the proper mails can get in the spam. So there was a suggestion from a previous groups that we actually communicate via Teams, and I will speak with other people who are running the classes how we can do it. And maybe actually it's a good idea, but at the moment, okay, let's stick to the sending by mail. If not, you will be notified by the leader if there are any other options. So please, in the mail subject, if you send by mail, you put your name, group, and report number. So this is it can be easily found in the the sea of the reports that we are going to receive. All the reports have to be sent in the PDF format. No, please do not send us reports that they are in the Word format. Okay, we are having different software, and the Word documents tend to actually not to look like you imagine them they look. So the PDF format is the most safe format. Please use Times New Roman 12 font, margins 1.5 centimeters from up, down, and sides. And as it comes to the space, please maximum 2 pages, okay, plus half a page for figures or tables, okay, and please use a single spaced written text. On each page, right upper side, please write your, again, name, surname, group, and report number. And very important, please insert page number. When we are printing your reports, and we are printing many, if there is the jam in the in the printer, then actually we get sometimes some of the pages mixed up, and just putting the page number and your name on the top of each page and the report number, it really makes our work much easier. So what you are supposed to put into the report? Title, of course, and aim. Remember that aim is not the title of the exercise, and the aim is short, as 1 or 2 sentences are absolutely enough. Then it comes the introduction, and it also should be short and concise, and usually what you can put in the introduction is why, what for given method is used in the molecular diagnostic lab. Then materials and methods. So I know from the leaders of the different courses that every single labs have special requirements as it comes to materials and methods. We here at the molecular diagnostics ask you that materials and methods are short. Please do not rewrite the instructions, okay, and do not rewrite methodology and or describe the description of methodology. So we do not need to have any buffer compositions, quantities of the used buffers or reagents, okay? So if you have trouble in deciding how to use and write materials and methods, look at any abstract at the publication, then it's a good example how shortly methods and materials should be written. Then results. Results have to be described. It's not enough to put just table or figure as a result. You have to describe what you actually have obtained as a result. Okay? Remember that all the figures and tables should be appropriately labeled. Also, if you have any doubts how it should be done, please go and look in any publication and see how the tables are labeled, how the figures are labeled as well. In the discussion, what you should put, you of course have to comment the results that you have obtained. So was the experiment successful, and are the results expected? You have to explain any deviations from expected results, and you have to discuss from where they may came from. Of course, what you can also include is what could be done to improve the quality of the result. So, for example, if the isolation of DNA resulted with the DNA with high presence of contaminations, for example, of organic material, what you could do to improve its quality. Very important thing, which is not written here actually, is language. Please work on your scientific language, and it is not. It's maybe not easy. You are not all very experienced in writing. So, very good exercise is to collect the vocabulary from a publication to actually find a good vocabulary. And now I'm going to address the Erasmus student and our foreign students that for whom the actual English, writing in English, it's very difficult, or maybe it's not that easy as for the grown-up scientists. You have to work on your reports. I imagine it won't be easy, and I will send you the links to the very good web pages where you can actually find good instructions of how to structure the scientific report. And for all of you, this information will be given. There is one excellent web page. It's called Writing Scientist. There is a blog of a professional who actually gives excellent tips on how to deal with writing, how to structure sentence, how to choose the sentences, and how actually improve your writing. So you will be given the information where to go to look. And again, look at the publications. If you have the problems with the sentence structure, rewrite good sentence from the paper and try to, let's say, change it to this, what you need to write. We have many students from, let's say, Roman language groups, Spanish, Italian, or French, where the sentence structure is totally, maybe not totally, but very different from the English sentence structure. So please make sure that you actually understand how the report should be written, how good scientific English for the communicating the results look like. What is absolutely to avoid is to try to Google Translate from your mother tongue to English. Generally does not work very well. If you can choose simple sentences, it's always safer than writing long ones, okay? I will give you in the at the end of the classes, I will send you all of the outlines for writing the report and the web pages where you can actually, get some help in scientific English scientific writing and scientific writing in general. Okay, and do you have any questions regarding grading, entry tests, or reports so far? Polish students, I think generally we always did like this, that you do it as it pleases you. So, but generally, most of the students in the, let's call it Polish language groups, they have a choice, so they can write in Polish, or so you can write in Polish. It's not a problem. Also, there was a question on the previous meeting about the exam. During the exam, you are going to have the question in both languages, Polish and English, and you can choose in which language you are you are writing. So just only that if you start to write in English, please write all in English, and so on and so on. Any other questions regarding grading, entry test, and reports, and organization maybe? No? Okay, so let's take 5-minute break, okay? And then we are going to continue with rules and safety measures while working in molecular diagnostic lab. with rules and safety measures while working in the molecular diagnostic lab. So these rules apply to the molecular diagnostic lab, as I said, and in some extent and with some little changes to the laboratory where you are going to work starting from next week. So your safety comes first. What is very important is proper hand washing and disinfection, bringing clean lab coats, wearing gloves, okay? Then proper shoes. So please do not wear any high heels. And although we are not in the summer season, there are no open shoes like flip-flops or sandals actually recommended to be used in the laboratory, any laboratory for that matter, okay? For people who has long hair, please tie them in a knot or whatever, so hair has to be up. We are not bringing to the laboratory any food or drink, we are not chewing gum, and we are not bringing any unnecessary things. And this is in the molecular diagnostic lab particularly important because it avoids, first of all, the contamination of the samples with your own material, so the material of the operator, and also the contamination of the things like, for example, calculators or phones that you are going next to take out from the lab and bring home. Important thing is to locate the fire exits, and when we enter the lab, this is one of the things which we will pay attention to. Okay, generally in the molecular diagnostic labs, as any other labs, there are several important labels that you have to be aware. You have to locate the emergency shower, emergency eye wash station, first aid kit, and also all the indications how to move in case of fire. And be aware that on our department we have these drills of fire drills quite often. It may happen during your lab classes that you will have to evacuate, so that's why we are going to remind you how to do it safely. Another important labels in any laboratory and laboratory of molecular diagnostic is the label of radioactive material, corrosive material. For you, especially important during the classes is where the biohazard material should be stored and utilized. Attention also to potential toxic material and to electrical hazard. And during the laboratory classes, sometimes you will use corrosive material like, for example, acids or alkali and some toxic reagents as well, for example, phenol chloroform. So proper labeling of the working space where you can actually work with this material will be indicated. So in all of the molecular diagnostics labs and any others, you have to wear your lab coat, and attention, the lab coat has to be closed, okay? Very often we use eye protection, so the special glasses in order to prevent the spill and of the material and getting potentially infectious material into eyes. This is, for example, a requirement, for example, when you are taking the samples out of liquid nitrogen. Of course, hand washing is necessary step, and the important thing is in the laboratories that the fire doors are kept shut during normal time, and they are only open during the evacuation time. So now I will tell you some words about the proper hand washing, and of course in the time of pandemic you are especially aware how important it is to keep your hands clean And actually, in the laboratory, the proper hand washing helps a lot, and is an important step. And now, just to remind you how to properly wash hands, you will see the WHO recommendation on hand washing. Just a moment. So you have seen this reminder how to properly wash hands. Just be aware that any jewelry is not recommended, as well as actually the artificial fingertips are also, not recommended, because they are usually longer, and this can afterwards lead to the tearing of gloves. Another thing which you have to check always while entering, washing your hands, whether you have any injuries. So if you do have, please put the band-aid in order to protect yourself. Okay, so why we are actually washing hands in the laboratory and in the molecular diagnostic laboratory? As you are well aware, on our skin we have transient and resident flora that is present, and what we can find on the skin surface, apart from the bacteria, there are some viruses, fungi, and also these little animals. It's not pleasant, I know, but they are mites that actually are our companions on the skin. As well. So we have several surfaces of the skin, as you are aware, and actually not only very external layer of the skin is habited by a specific skin microbiome. Actually, this bacteria and some viruses can go a bit beneath this first shed of the skin epithelium. Okay, we have also bacteria present in the sweat glands, and in the hair roots as well. So the skin, what is present and how we are colonized by different microbiome species depends on sex, age, and site. On climate, of course, geographical location of on our immune system, and possible pathologies that we have. Of course, lifestyle, and so what we do and how we keep our hygiene, but also on host genotype. So here what you can see is how different locations in our body are colonized by different microbiome elements. And of course, it varies depending on how oily the skin is in a given place, oily, wet, or dry, or whether it's exposed to light or whether it's hidden. So here, this is the example of the skin colonization in a different places in 4 different healthy volunteers. So you see that the composition of this flora can vary a lot in the individuals. However, what you find most prevalently on the skin flora, there are Staphylococcus epidermis and Propionibacterium acnes. Other elements of the skin flora include some fungal species. This is Malassezia mainly, and also the Modex mites. And the proper hand washing technique allows you to get rid of some of the transient flora from the skin. And this is very important while working in the laboratory and also for your own protection. So once you have properly washed your hands while you enter to the lab, what you have to do is you have to put the gloves, and then after working, you have to take them off. So how actually we safely remove the gloves, you will see just in the moment.
I'm an infection prevention specialist at UT Physicians. Joining me today is Jeff Lindsay. He is a nurse with UT Physicians, and we're going to be showing you how to don and doff, or put on and take off, gloves. Go away, Jeff.
Before donning gloves, always perform hand hygiene. If you have any questions about hand hygiene, please see the related video. Take a pair of gloves from the glove container. Make sure to select the correct glove size. Place fingers into the first glove and pull the glove onto that hand. With the non-gloved hand, place fingers into second glove and pull the glove onto that hand. If you notice any rips or tears in the glove, then replace the glove. And remember that if you're wearing a gown, the gloves go on top of the gown. And for doffing gloves, the outside of the gloves are contaminated. Using a gloved hand, grasp the palm area of the other gloved hand and remove the first glove. Hold the removed glove in the gloved hand. Slide fingers of ungloved hand under the remaining glove at the wrist and peel off second glove. Discard the gloves. Remember that gloves do not replace hand hygiene. You must perform hand hygiene when taking off gloves.
Okay, so we continue. So now you know how to wash your hands, how to put and remove the gloves safely in the laboratory in order to avoid actually contaminating yourself if your gloves has been contaminated. So when we speak about the contamination, you have to be aware, and this is important, is that any material that enters to the molecular diagnostic lab is potentially infectious. And not only potentially infectious, we tend, as molecular researchers, we actually work with the material about we know that it's infectious. However, any material, even if comes from the let's say blood bank, is treated as potentially infectious. So of course all the precautions measures has to be taken in order to avoid the risk of disease transmission on manipulating personnel. However, your safety is the foremost issue. However, we cannot forget about the possibility of cross-contamination of samples, so in so-called carryover. So the samples can be contaminated with material coming from manipulating person or coming from another sample, and that in the molecular diagnostic is especially dangerous, and as it leads to the false positive results of the tests. So what happens, how to organize the work in the lab, apart from the safety measures and preventive material like wearing lab coat, gloves, and so on, is to separate the isolation of nucleic acids and analysis, so by space and time. And generally, as in molecular diagnostic nowadays, we base very often our analysis on the PCR, is to separate pre-PCR and post-PCR areas. And so the pre-PCR sample preparation, like nucleic acids isolation area, should be separate from the PCR setup area, which in turn has to be separated from the post-PCR area. And this is how good laboratory should be organized. So we have the sample reception at one place where the sample is received, where it's proceed until the nucleic acids are isolated. There is a separate place where actually only clean reagents for the PCR setup are mixed. Then there is a place where where the PCR mixture and the matrix, so DNA or RNA, are mixed together, and then they are going to the instrument room when the PCR is run, and the tubes with amplified material are open and analyzed, for example, by electrophoresis. So, of course, in the conditions of working in the didactic laboratory are not as such, and what we tend to do, we will first isolate these steps, but by doing isolation of nucleic acids at the different time. So this is the first thing. The second thing, we will choose the areas in the lab, which will be the clean area, like the PCR setup, and we will analyze the results. So prepare the agarose gel and run the gels in the separate bench in order to avoid the contamination by amplicons. So this is about the space. And as I told you, the clinical material which we are working for, they are all potentially infections, and apart from blood, which can be whole blood or purified leukocytes, PBMC, serum or plasma, we can have also sputum, the liquid from bronchial lavage, oral cavity, and pharynx. We can have as a starting material the cerebral spine fluid, urine, feces, swabs from the genitourinary tract, pieces of tissues coming from biopsies. We can have also archival material, tissue fixed in paraffin blocks, for example. We can have archival material like skull, bones, or teeth. We can also isolate material from placenta, for example, or the pre-implantation embryo in case of prenatal diagnostics. We, of course, can work also with very little amounts of the material, like spots of blood or semen, and all of this material is considered as potentially infectious. So what are the criteria for accepting specimen for testing in the molecular diagnostic labs? They are the criteria that allows for traceability of specimen and traceability of the information and all the data that comes with it. So, of course, if the sample is compromised in some way, for example, broken tube, or it has be always noted. All of the chain of the working chain and proper result is guaranteed by a proper labeling. And of course, we are not in the times when on the tubes there was a name written, or now we are operating with labels, barcoded labels, so, and a special equipment that actually allows proper traceability. Of course, what has to be always noted is the time and place of collection of the material, for what it is, very often contact to a physician, and all the necessary patient consent forms. Also the information about the ethnicity and medical history that is relevant to the analysis of the specimen. What the precautions in sample proceeding has to be performed. Of course, as I told you before, all the specimens are potentially infections, so we wear personal protective equipment, and of course we are not only about to protect material, but ourselves, but also material. So we protect the material from degradation and cross-contamination. We generally we have we separate RNA and DNA handling. All of the holding and storage requirements must be met, like the verification of the temperatures in the freezers and fridges. And they are for the blood, bone marrow, or some of the sera or plasma. There are the special requirements that has to be met at transportation, at the reception of the samples. Of course, frozen tissue has to arrive to the lab frozen, and we thaw it at the moment when we are ready to isolate the material for the analysis. Few words about the potentially infectious agents that are present in the clinical material. So, of course, we are well aware about the blood-borne viruses, BBV. So we are having hepatotropic viruses, which are hepatitis B virus, hepatitis C virus, also hepatitis G virus, okay? And of course, the viruses of the acquired immunodeficiency, so HIV-1 and 2. Be aware that apart from blood-borne viruses, we have also the viruses which are vector-borne viruses that can be actually carried out by blood-sucking animals, such as mosquitoes, ticks, and fleas. And there are several viruses and several parasites which can be transmitted in this way. This is like West Nile virus, Zika virus, dengue, yellow fever, tick-borne encephalitis, and malaria, just to give you some examples. Of course, during your laboratory classes, the first experiment you are going to perform is on the DNA isolation from the blood, and you will be given the blood from our local blood bank. And this blood that you are going to work with has been tested for all known pathogens and viruses. So, however, we always treat this material as potentially infectious because it has been checked for known pathological agents. However, we cannot be sure that, let's say, in a couple of years, some hepatitis Z virus will be discovered, and it may appear that you had the contact with it before. So any material that comes for the analysis, we treat it as potentially infectious, okay? So as I told you before, protect yourself always, label tubes appropriately and all the containers. And it is believed that any biological material is dangerous until after the lysis is performed. what you have to do always, you spin down the tubes with blood or any fluid that is your starting material to avoid spill from lids. Okay, and afterwards you inspect your workspace, your hands, gloves, pipettes, and table, and you see whether or not there is any contamination that it's present. Even if you do not, of course, notice an obvious contamination, you always clean the bench after working with the biological material. Okay, so what you should do if something happens, like you have the spill of the blood, okay? So, and also if, I don't know, you have the spill of some toxic reagent or radioactive reagents or corrosive reagents, okay? So one very important rule to apply is you do not panic, okay? So you do not move chaotically. You avoid to any rapid movements. You stay in place. And first important rule is verbal information. Okay, attention, I need help. Blood is spilled. So we have noticed during the lab classes that we had recent years that very often when you are giving blood, you tend to be nervous. And actually, it's a good thing. You do not have that much experience. And sometimes your hands are trembling, you are a bit afraid, and it may happen. So please do not panic, okay? It may happen. You will know how to react. Okay, so this is the first thing. The second thing, when you spill any of the material that it's potentially dangerous, biologically dangerous or otherwise, you do not wipe. Okay, wiping actually leads to spreading, not to cleaning. So first of what you do, you pour disinfectant over the spot or the spill, okay? And about the disinfectants, I will tell you some words afterwards, because the disinfectant in the molecular laboratories molecular diagnostic laboratories, while working with the potentially infectious material, has to be chosen very wisely, okay? So you take disinfectant, you spray or you pour it over the spot, okay? And then you put absorbent towels on the stain, and then you wait. How long you wait, it will depend on which disinfectant you are going to have in the lab, okay? Then you throw absorbents away and repeat all these steps until there is no blood remaining and the surface is clean. Then you dispose the material, the contaminated material, to the appropriate waste bin, and just remember, this is the sign of waste bins, which should be on the waste bins where the biohazardous material has to be thrown away. Sometimes these containers are red, but they do not necessarily have to be in this color. The most important stuff is just this sign of biohazard, which also you can find if you move around our building in any floors when you enter to the working space, because it's also the sign that tells you that you are entering into GMO working area, okay? Afterwards, you change gloves always. It's a good practice, and you change lab coat if it's contaminated, and if you have contaminated with the sample. So I have mentioned to you that disinfectants are choosing wisely disinfectant in the molecular diagnostic lab while working with the biological material that is potentially or actually not potentially, but it's infectious, is an important step. And very often in the molecular biology or genetic engineering labs, what it's used to for the decontamination is 70% ethanol. And while working quite well, while working in these labs, in the molecular diagnostic labs, actually it is not enough. So what we are using, generally we are not using just ethanol. We are using the mix of alcohols. So and the most often we use ethanol mixed with isopropanol, and this is the example of this disinfectant. It's called Incidin. It works very quickly when the blood spill is not very big. It actually performs its work in 30 seconds. And it also shows quite a wide spectrum of working toward various bacteria and viruses, okay? Including, for example, hepatitis B or virus or the tuberculosis bacteria, okay? Then you can have also the mixtures of alcohols and aldehydes, mainly glutaraldehyde, okay? It can work at 30 to 60 seconds, depending also on the amount of the material that has to be disinfected. Also, it shows quite wide spectrum of working. Another example is the Medicept. It has alcohols, aldehydes, and some other reactive reagents that help to disinfect, and also has the wide spectrum of action. And if you then compare this wide spectrum of action with the just 70% ethanol, it comes that in order to eliminate most, but not all, attention of the potentially infectious agents, okay, you need 10-15 minutes. And the spectrum is not actually very wide. It is known that 70% ethanol is not sufficient to remove, for example, hepatitis B virus or tuberculosis. It may, however, change a bit with ethanol concentration and the time of exposition. And here you have the example, you see at the ethanol. So what can be used, it's from 70 to 85% of ethanol. However, it's not a broad spectrum work, okay? It works well toward normal vegetative bacteria. Again, it works quite okay against some of the lipoviruses. On the non-lipid viruses, it depends on the concentration and time of exposition. It does not work at all toward the bacterial spores, and it not always work toward all of the species of Mycobacterium tuberculosis. So here you see actually that how choosing a proper disinfectant in the laboratory is important. So what you should know from the previous classes on microbiology is that disinfectant does not mean a good antiseptic and vice versa, okay? And that the antiseptics, okay, generally has to be a bit milder because they are acting on the skin directly, okay? Another thing which you have to know is what is the difference between the disinfection and sterilization. And about this, I will send you some material to read afterwards. Okay. I have told you something about the glove selection, about the wearing gloves, and be aware that the glove selection, apart from the selection of the disinfectant, is also important. So generally nowadays we tend to avoid the latex gloves and powder gloves and we use nitrile gloves, and it's quite okay. However, if you are going to work at the same time with highly corrosive material or with some of the toxic material, the normal nitrile gloves may not do it, and I will send you a list of how actually the material from which the gloves are done should be taken into consideration while working with some other toxic material. What is important in the molecular diagnostic lab and any laboratory that takes and works with infectious particles is let's call it mixture of gloves and disinfectant. Okay. while 70% ethanol evaporates very quickly and may not do much of the harm if you spray your gloves with it. If we come to the mixture of alcohols and aldehydes, okay, then we start to have a problem, because this is actually harsh on the, it can be harsh on the nitrile gloves and not only nitrile gloves, and it leads to creation of some little micropores. So generally, what should be avoided in the laboratory, but it's a common sin of many of the researchers, scientists, and students, that we want to work clean. So we put the gloves and we put the disinfectant over it. And actually, while working with the biologically infectious disease, the material, it should be avoided, okay, because of what I told you before. While working with the high volumes of blood, or with the blood which we know that it's surely coming from a patient that, for example, has been infected with the HIV virus, or, for example, if you are working in the biosafety laboratory 3 and you are actually making viruses, lentiviruses or retroviruses for your work in order to introduce a gene into the, I don't know, the cells, it is recommended and it is necessary to wear 2 pairs of gloves, the internal and external one, and the external one in these cases should be changed more or less every 20 minutes. So this is something you should be aware of as well. Okay, and we will take now another 10-minute break. Okay, and because my voice my voice has to rest a moment. And during this time, okay, I will send you now all the materials that you need to have for the classes. Okay. Do you have? But before we stop for 10 minutes, do you have any questions that that comes to your mind regarding safety in the laboratory? No, okay. So we make 10 minutes break. About to finish in sometime soon, so you're happy about this and me as well. And do you happen to have any more questions? No, so I will continue. Yes?
I just wanted to say that I don't have any questions.
Okay. No, the reports are written individually. Okay, this is the important thing. So everyone has to give the report attention, and it will be given that you are not generally, of course, you write about, let's say, your experiment, but it's well seen, let's say, if the whole group did not succeed on the DNA isolation, okay, or 10% succeeded, okay, to write it in the report, okay. So also, for example, when you are going to write the report on the genotyping, you will have to analyze the result of the whole group, okay, because this otherwise makes no particular sense. But anyhow, all the information will be given to you at the classes, okay. But yes, report you are writing by yourself, so be aware about copying from other people or from other groups. We actually see it quite immediately that we have, 2 very similar or identical reports. Okay. Any more questions? Okay. So next. We are going to do some more stuff and then it will be over. You will have actually the time afterwards to prepare for your first class. But before you do so, okay, sorry, some more information for you, okay. So how to avoid the cross-contamination of samples, okay. This is a very common rule in all of the labs, that you have to spin down all the samples, reagents, buffers before every use, okay. Of course, once again, you wear gloves and attention has to be paid to do not touch your face or hair, okay. You are going to have gloves, okay, and luckily nowadays you are going to have masks, so then actually it prevents from touching. But anyhow, I know, and we have noticed it, that you are not that much experienced and you still may have some problems with controlling your let's call it proper behavior while working. So we are going to pay special attention to this during the classes, okay. But be aware, you have to start to put the good habits in place, not touching anything apart from laboratory devices during the lab, okay. Very important thing is to well organize your space in order to avoid contamination, okay. And I will show you how we do it properly in the next slide. You have to be aware that all of the tubes has to be labeled properly, okay. It's not enough to write on the tube that it's a DNA. In the common box where you are going to store the material for the classes, you will have like 20 DNA samples. So you have to write the date of the isolation, from whom it is, or the number, whatever. You have to be sure that all of the tubes you are going to use and the material and so on is properly labeled. And another thing is to work smart. And in order to work smart, you have to well organize your time and space, okay? So very soon you are going to go to the lab, not only to the classes, but to do your master thesis, for example, or to be in the lab because you have the opportunity to do so. And how you prepare actually before, it reflect and may reflect quite a lot on your performance and how well you actually do. You have to, as I told you just at the beginning, you have to know what you are going to do, how you are going to do it, and what it's necessary. So what kind of the reagents you are going to use? Do you need to thaw them, remove from freezer, fridge, and prepare before, okay? What kind of equipment you will need during the work? For example, while you start working, you have to ask yourself, do I need some equipment that has to be specially prepared before, calibrated, for example, pH meter? Or whether very often in our classes, you will need thermoblock afterwards at high temperature. So make sure that you switch on all the equipment that you may use afterwards. And sometimes, we are giving you the instructions that follow certain flow. However, you can prepare some things in advance. For example, the gel, because there is a time for setting the gel, like half an hour. So it's sometimes better to switch the steps in order to gain some time and better organize yourself during the classes. That's why it's absolutely necessary that you read the instruction and know what are you going to do. Okay, and now we come to the space organization. So organization on the bench and organization of the working space. Crucial, we do not bring in the working area any unnecessary equipment because of the possible contamination that may occur. Okay, and how do we organize ourselves? We keep two distinct, let's say, spaces, or actually even three, in the front of you. Okay, so if you imagine yourself in the front of the bench, okay, you have your left hand and right hand. and you have your right part of the bench and left part. So in front of you, what you will have your working or operation space. So actually where all the mixing, aliquoting occurs, okay? On your left, on the top, you should have always clean material. So let's say, and also some pipetting devices like pipette, okay? This is really keeping, keeping clean stuff separate from the used stuff, dirty stuff. It's crucial to avoid the contamination, okay? Cross-contamination and contamination. In front of you, generally you put all the reagents and the test tubes just in front, okay? So how you are supposed actually to work? You take your right hand, you take a pipette, you put the tip, okay? You aspirate, you take whatever you need, you open the test tube at this moment, okay? You add it, and then you discard the tip, okay? So you have, when you have the, so actually the direction of working is a bit like this, okay? So diagonal. From this, okay, pipette, tips, aspirating, putting, throwing, okay? Sometimes the waste bins actually can be placed, somewhere here, okay? Or sometimes when they are huge, big waste bins in some laboratories, there is a common usage of the waste bins which are open and are placed on the floor. This is not the case, okay? We keep everything on the bench, however, in the right position. Important thing while working, okay, we do not leave any of the tubes or reagent bottles, containers open. Okay, we open them, we take what we need, we close them. Okay, so this is very, very important. Of course, if you happen to be left-handed, then you organize other way around, okay? Sorry. And you work like this. Actually, having left-handed people in the lab, it's not problematic, but it's always that these people, have, let's say, different organization of the space. Okay, so you change it whether you have a colleague or partner who is left-handed and whether you are right-handed, and this is something that is maybe not so easy, but actually it's doable. Okay, so organization of your space is crucial, and actually we will be looking at you during the classes and we will be correcting you in order for you to take the habit of proper working. And this is true in any laboratory, molecular diagnostics, whenever you handle specimen or samples, this works like this. Okay, and when we speak about the organization of time, space, and actually working, manual skills are important. We are not surgeons, luckily. However, having the good manual skills helps a lot. But then, luckily, with experience, you will gain it. Very important thing in the lab are pipetting skills. Okay, it happens to all of the researcher who has a break in the lab, and for example, me, when I write a paper, I do not enter the lab for a long time. What I start by doing is I go and to remind myself how to properly pipette, okay? You were very unlucky recent years because of the pandemic. You did not have many of the classes. You did not have so much exposure to the laboratory environment as usually students have at our department. That's why on the first classes we will practice pipetting skills. And pipetting skills are very important in order to properly measure and add reagents, and you will see how good your pipetting skills are during when we are going to do the quantitative PCR. Okay, any quantitative measurements and analysis are very susceptible even to the small, and quantitative PCR is a perfect example of it, where the small mistakes in the pipetting reflects in the obtained results. And now you will have 15 minutes in order by yourself to find good YouTube video or Bing video on the pipetting skills. So what you are supposed to remind yourself is how to set the pipette, okay, for the desired volume, how to aspirate the fluid, different fluids, viscous, for example, or the fluids that are not viscous, like alcohols, okay. Then how to do the releasing of the liquid, okay? How deep the tip should go into the liquid, okay? And how to do repetitive pipetting, okay? So this you are going to do in the end of the classes, okay? You will find yourself the good video, and you are going to watch it, and we are going to speak about the pipetting and how you do during laboratory classes. What you also will do now, you will remind yourself about the calculations in the lab, okay? As I told you, you are not going to bring calculators, okay? This is in order to avoid the contamination and taking it home, but also this is necessary to train your mind and your brain. We are not going to do very sophisticated mathematical calculations. They are simple, you know, changing of recalculating the concentrations. This is one thing you have to know, and they are the examples which you have to know how to put the same, how to put the one concentration into the another one, okay, that corresponds to it, okay. So this is the example. This is something which surely will appear during the entry test, okay? So in the lab, we are working with microliters, milliliters, or liters mainly, okay? We do not have any equipment. We are not equipped with the pipettes that allows you, for example, to pipette nanoliters, although we have some of the machines that can do it, okay? From other side, we have micrograms, milligrams, and grams. So any kind of conversion between these concentrations, you have to know how to do it quickly. Also, in the lab, very often when you are working, there are restricted areas, like, for example, BSL, where you should not bring the calculators. And then, of course, for the mind training, is you have to know how to do it. If you have some problems like dyscalculia or something, please let me know, and the proper physician certification is necessary for this, so that it can be, of course, the exception. And as making mistakes while not using calculator, well, if you know how to do it, you know how to do it, and you can do mistake both ways, whether you work calculate with your pen, pencil, and a piece of paper, or when you press the buttons on the calculator. Okay, so if you do not know how to do it, you can make the mistake anyhow. Okay, so this is the example of what you should know how to do. Another thing what you are supposed to know how to do is to do the simple recalculations. For example, like this one, which is necessary for setting up the PCR reaction. So, for example, when you are given certain stock solutions like buffer, magnesium, DNTPs, primers, polymerase, and template, when you are giving the final concentration of this, okay, so the question is how much, so what volume of each of these reagents you need for this final concentration for given volume, which can be different, okay? So Now you have time actually to practice a bit, see, find the pipetting skills video, see whether you are familiar with the calculations, okay? And if because we have still more than 1 hour, if you have time, please start preparing for your entry test next week. Okay, and I will meet with you, let's say, 5 in 1 hour, okay, just to ask you whether you have any particular questions. And if not, I just leave you with your little work to be done now. Would you like to ask about something now? No? Okay, so I leave you for 1 hour and let's meet 4 for our final time, okay? See you soon. Work well. Use this time, okay? Because you use it now, you will have to, spend less time preparing for the entry test and for the classes.
Halo?